Hey guys, welcome to Young-Splaining Cell Division Mitotic version. So, in order to first even understand what cell division is, we need to understand why we need cell division. And the purpose of cell division is to maintain the continuity of life. Now, there's three ways this is done: through reproduction, where genetic information is passed from one generation to the next; growth, where an organism has the ability to increase in size as it goes from infancy to adulthood; and repair, where the organism possesses the ability to make repairs to damage that is done throughout its lifetime. Now, after knowing why we need cell division, we need to know how cell division occurs. And the first thing is to understand the structure of a cell. All cells, whether they're prokaryotic or eukaryotic, have a plasma membrane, cytoplasm, ribosomes, and genetic material. Now, the genetic material of prokaryotes is going to be different than the genetic material of eukaryotes in the way it is contained. Prokaryotes tend to have a single circular chromosome, whereas your eukaryotes are going to have two or more linear chromosomes. So let's look at the difference in the way that they're actually packaged. Now, DNA molecules are packaged into chromosomes, and we know that DNA is simply a sequence of nucleotides, the adenine, thymine, guanine, and cytosines that are held together with those sugar phosphate bonds in a double helix formation. Now, DNA itself, a simple nucleotide, is only you have the sugar, which is going to be deoxyribose, a pentose or 5-carbon sugar. You have one of the four bases I just named: adenine, thymine, guanine, and cytosine. And you also have that phosphate group. The sugar and the phosphate, again, are instrumental in forming the backbone. And the bases pair to form like the steps, if you want to call them, of the double helix, which kind of looks like a spiral staircase. Now, eukaryotic DNA is going to contain its entire genome. All of its genetic information, all of its genes are found in a single circular chromosome. Some prokaryotes actually have small pockets of DNA called plasmids, so that if that prokaryote is in a bad condition at the time where the environment is not conducive to its survival, then it is able to keep genetic information safe in those plasmids, or to transmit those plasmids to other bacteria to pass on genetic information until conditions are more favorable. Eukaryotes, on the other hand, consist of a number of DNA molecules. For example, the human genome consists of 23 pair of chromosomes. And that 23 pair, we have 46 chromosomes, two for each chromosome, for each type. Now, the chromosomes in eukaryotes are not just the DNA like we see in prokaryotes. In eukaryotes, there's DNA and protein, specifically histones. Think of histones as being like the spool, and DNA as being like thread that can wrap around or unwind from that spool. When the cell is not undergoing the cell cycle, when it's not reproducing, the DNA is going to be in a relaxed form called chromatin. It's not visible with a light microscope at this point because it is so thin and so stretched out. Now, we have two types of cells in eukaryotes. We have somatic cells, which are the body cells, and somatic cells tend to be diploid, meaning that they have two copies of each chromosome. N is the letter that represents the number of chromosomes. So if you're diploid, you have two copies of each, 2N. Remember I said humans have 23 pair. So the number of chromosomes we have would be 23. We have 23 different types of chromosomes. There's two of them for each chromosome. So that means that our diploid number is 46. Our gametes, or the sex cells, oocytes, which are eggs in females, and spermatocytes, which are sperm in males, there's only one set of chromosomes. So we said those are haploid because they have half the genetic information of a somatic cell. So there's only one set of chromosomes. There's not two sets. So they're haploid. Now, looking again, remember that the prokaryotic chromosome is going to be a single circular chromosome, whereas our eukaryotic chromosome is linear. Now, what we see here is a replicated chromosome. This is one chromosome that contains two strands of identical DNA. These are the chromatids, the sister chromatids. So I know I've said something about sister chromatids and having two pair of chromosomes for each, two chromosomes for each pair. Well, that's because they're homologous. So in a eukaryotic organism, the gametes fuse through fertilization to restore the diploid number. Now that means that you get one complete set of chromosomes from your father and one complete set of chromosomes from your mother. So you have paternal chromosomes and maternal chromosomes. Now these chromosomes would be considered homologous to each other when they carry the same genetic information. So let's say that this was chromosome 1 of our 23 pair of chromosomes. If chromosome 1 contained the genes for eye color, hair texture, and height, then the one from dad would have those 3 genes, and so would the one from mom. So this is a pair of homologous chromosomes because they contain the same genetic information. Now, during the S phase, which is a part of the cell cycle, the DNA, the genetic material, is going to be replicated. When that genetic material is replicated, each chromosome makes an identical copy of itself. So here you have 2 chromosomes and 2 chromatids. So there's only 2 strands of DNA for those 2 chromosomes. Here you have 2 chromosomes. So the paternal chromosome is still homologous to the maternal, still considered 2 chromosomes. However, there are 4 pieces of DNA or 4 chromatids. These 2 are considered sister chromatids because they are held together by a centromere and they are replicated chromosomes. So here are the maternal sisters, here are the paternal sisters, and the paternal sisters are non-sisters to the maternal information. They're homologous. So sisters, sisters, non-sisters, or homologs still carry the same information. So to go over it again, 2 chromosomes, 2 chromatids, 2 chromosomes, 4 chromatids, okay? Homologs carry the same genes, one received from each parent, and sister chromatids are the result of DNA replication held together by a centromere until anaphase. Now, eukaryotic mitotic cell division is going to take one parent cell and produce 2 identical daughter cells. Each daughter cell is identical to the original parent cell. For the sake of teaching purposes, we always use a diploid cell to do this. However, mitotic division can happen with diploid or haploid cells. We see that with the haploid cells in the alternation of generations with plants. Now, the point is mitotic cell division is going to result in genetically identical daughter cells. There's 2 main phases of the mitotic cell division, and each of those phases has a subphase. Our first phase is a preparation phase known as interphase. We spend more time preparing than we actually do dividing with the cell cycle. So interphase is the longest phase of the cell cycle. The second phase is the actual division of the cell. The division of the cell happens in 2 main parts. First, the nuclear information divides, known as karyokinesis or mitosis, and then in most cells, the cytoplasm will divide. This is known as cytokinesis. Not all cells undergo cytokinesis. And when this happens, in this case, like we see in smooth muscle, we end up with a multinucleated cell. Okay. So let's look at our preparation phase or interphase. Again, interphase is the longest part of the cell cycle. The cell spends about 90% of its time in the cell cycle and is broken into three subphases, G1, S, and G2. Now, the cell grows throughout all of interphase, but it has significant growth in the G1 and the G2 phases. So, G1 phase is the first gap or gap 1, often referred to as the first growth. This is where the cell starts to grow at a faster rate than normal. In the G1 phase, there is also a G1 checkpoint. That G1 checkpoint is considered the most important checkpoint of the cell cycle because cells that go through G1 tend to commit to the cell cycle. Any cell that cannot undergo G1, that is not going to make it past that G1 checkpoint, falls into the G0 or the non-dividing time of the cell where it is not going to undergo cell cycle if it is not damaged, or it may be signaled to undergo apoptosis, programmed cell death, if there is some damage. The G1 checkpoint is checking to make sure the DNA is not damaged and that the cell has not reached its Hayflick limit, or the number of times it can divide safely without causing damage to the DNA or loss of genes. Following the G1 phase is the S phase, or the synthesis phase. This is the phase where the chromosomes are actually going to be replicated. This is the time when the genetic information is doubled. Now, even though the genetic information is doubled, the chromatin is still relaxed, so you can't see the chromosomes yet, but this is where the genetic information doubles. The S phase is followed by G2, or the second gap phase. This is where more growth continues. You're going to see things like your important cell organelles and your centrioles being replicated here. And there's also a second checkpoint. The G2 checkpoint is going to be checking to make sure that each chromosome has been replicated, that the DNA has been replicated at this point. During the cell cycle part of interphase, there is a signaling factor called MPF, mitosis maturation promoting factor. And this factor is building up, and those levels have to get to a certain point before the cell can also progress into the M phase. So in addition to these checkpoints, in addition to the synthesis happening, you also have internal cell signaling, where you have a chemical signal that is building up in levels so that the cell can actually go on to the next part of the cell cycle. The next part of the cell cycle, phase 2, is the actual cell division. Now, the M phase, or mitosis, is where you're going to get division of the nuclear information. Now, M phase occurs in 5 subphases called the PPMET: prophase, prometaphase, metaphase, anaphase, and telophase. Now, during prophase, this is the first part of the nuclear division, and in order for the nuclear information to divide, because the DNA is contained inside of a nucleus, one of the first things that has to happen is the nuclear membrane has to break down. So the nuclear membrane, which is the set of phospholipids, that phospholipid bilayer that actually separates the genetic information from the cytoplasm, begins to disintegrate. The nucleolus disappears. Your chromatin is going to condense to become actual chromosomes. So think of that DNA wrapping around the spool to shorten and thicken. This is the first time you can actually see the genetic information in the cell. Your centrioles, which have been replicated during interphase, will begin to migrate to opposite ends of the cell. As they're doing that, the mitotic spindle begins to form. The next phase is prometaphase. This is the part that is between prophase and metaphase. Now, during prometaphase, your centrioles will have reached the opposite ends of the cell. The nuclear membrane will be pretty much gone at this point. You'll still have little fragments of it, but it's pretty much disintegrated. As you see, there's really no nucleus left anymore. Reason being, these microtubules that are a part of the spindle will begin to attach to the kinetochores on these chromosomes. Now, each one of these sister chromatids is held together in the center by a centromere. On part of the centromeres are kinetochores. So there is a kinetochore on each side of each sister chromatid. Now, from your centrioles, those mitotic spindles, there's two types. You have kinetochore spindles and you have non-kinetochore fibers from that spindle. The kinetochore fibers are called kinetochore microtubules, and they are going to begin to attach to the kinetochores on either side of these sister chromatids. So if you see here, you see this sister chromatid has a kinetochore fiber connected on both the right and the left sides. This is going to be essential for lining up these sister chromatids for metaphase and for separation in anaphase. Now, your non-kinetochore microtubules are actually going to push against each other, and they push against each other to elongate the cell, to make that cell even bigger. Okay, so this is what's happening during the prometaphase. And prometaphase is essential for the next phase, which is metaphase. Now, metaphase, these microtubules, the kinetochore microtubules, are going to move the chromosomes back and forth until they get them in a straight line in the equator of the cell. This equator is known as the metaphase plate. If you notice, each sister chromatid is attached to a kinetochore fiber on both sides. This is essential. There is also a checkpoint. This checkpoint is called the M checkpoint. The M checkpoint is checking to make sure that every kinetochore microtubule is connected to a kinetochore on either side of these sister chromatids. When that is done, an enzyme known as separase is going to catalyze the cleavage of the centromere. So think of the catalase as helping with the scissors to cut these sisters. The connection that's been holding them together is going to sever that connection. When that connection is severed, the next phase is anaphase. Anaphase is a separation of sister chromatids. Now, on the kinetochore side, those microtubules are going to start degrading. I always think of the kinetochore as being like a pencil sharpener and the microtubules as being like the pencil. So it begins to degrade the microtubules, making them shorter. And as they get shorter, the chromatids are separated from each other and move to opposite sides of the cell. At the same time, the non-kinetochore ones are pushing against each other, elongating that cell, making it longer and longer, pushing out that cytoplasm. Following anaphase, you're going to have telophase. Now, telophase and cytokinesis often overlap. These phases often overlap with each other. The key parts of telophase is that it's the opposite of prophase. So anything that was done in prophase will be undone in telophase. So in prophase, the DNA was condensed. So it relaxes back out to chromatin in telophase. You can no longer see the chromosomes in telophase. The nuclear membrane begins to reform. So for the first time, you can see two nuclei in the cell because it's still one cell. The cytoplasm hasn't divided yet. So it's still one cell. And so you're going to have two nuclei in that cell. And as you see here, you have one pair of centrioles on both sides, as opposed to having two in the cell. The reason they migrate to opposite sides is so that each cell can have its own that will again replicate during interphase when the new cells get ready to divide. What you often see begin forming in animal cells is known as a cleavage furrow. At this point, this is the beginning of cytokinesis. If this was a plant cell, you would not see this. You would see a cell plate beginning to form. because plant cells and animal cells divide their cytoplasm differently. Now let's look at cytokinesis, which is the actual division of the cytoplasm. As I said before, plants and animals do this differently. Animals go from the outside in. There's a contractile ring of macrofilaments, kind of like putting a rubber band around it almost, that begins to squeeze on the cytoplasm, forming this, what we call a cleavage furrow, and it continues to pinch off until the cytoplasm is divided between the two daughter cells. And so you end up with two identical daughter cells, which were identical to the original parent cell, each with its own set of DNA. Each one of these cells go back into G0 until they are ready to divide, and then they go from G0 to G1, and they will go through the cell cycle again. When a cell is not dividing, it is doing its normal work. It is doing whatever it is supposed to do. If it's a skin cell, it's protecting. If it's a liver cell, it's detoxifying. So whenever a cell is not actively dividing, it is doing its job, its individual job. When it's doing its individual job, it is in the G0 phase. Here you see a plant cell. Plant cells divide the cytoplasm from the inside out. So what you're going to have with plant cells is you're going to get a bunch of Golgi that actually line up in the middle, and then they begin secreting the cellulose that is actually going to form a cell plate, which will ultimately combine with the external parts of the cell wall to make two new cell walls for those daughter cells. So plant cells divide from the inside out. Animal cells divide from the outside in. Now, let's talk about the difference between prokaryotic and eukaryotic cell division. Prokaryotes, because the genetic material is not contained inside of a nucleus, the division is much simpler. What you're going to have is the chromosomes will begin replicating at that origin of replication. And as the chromosomes replicate, the cytoplasm will begin to elongate. Once it elongates, as it elongates, the replication will finish and the cytoplasm will pinch off, kind of like a cleavage furrow, and you have two identical cells. This process is known as binary fission. Fission is a division. Fusion is when two things come together. Fertilization is a fusion, and this is a fission or a separation. So binary, meaning it goes from one cell to two. So binary fission. So now that we know how the cell cycle happens, let's talk about the things that trigger the cell cycle to happen. First thing is you're going to have internal and external signals. Now those internal signals were things we talked about, like that MPF, that's going to send a signal that lets that cell know that it's time for it to replicate. Your external signals, so you're going to have things like growth factors. And growth factors are like your hormones that can actually send a signal to the cell that tells the cell that it is time to make more. One of the things that controls the cell cycle is called density dependence. And density dependence is based upon the space the cell has available to grow. Excuse me. Think about when you get a cut, right? You're going to get a scar from it, but it's not going to continue to grow. Like once that cut heals, that's all of the space that the cells actually have to fill in. And once they run out of space, they receive a signal to stop reproducing, to stop going through the cell cycle. This is one of the problems we see with cancer cells. They do not obey density dependency. Another external signal is anchorage dependence. This is basically stating that a cell has to be anchored to something. It has to be attached to the cytoskeleton in order for it to actually undergo the cell cycle. Like the cell won't just be floating in the air and be able to reproduce. We also know that this is a problem with some cancer cells, especially with Henrietta Lack cells, because her cells could grow on anything and anywhere. They did not require anchorage to the cytoskeleton in order for them to actually reproduce. So anytime there is a loss of cell cycle control, that is called a cancer cell. Now, having a cancer cell and having cancer are two different things. At some point, everyone has had a cancer cell in your body. Typically, the immune system recognizes a cancer cell and gets rid of it. A normal cell can be converted through a cancer cell to a cancer cell through things like mutations or exposure to certain toxins. And that process is called transformation. So when a normal cell is converted to a cancer cell, it's called transformation. When the immune system does not eliminate a cancer cell and that cancer cell continues to divide, it forms what's known as a tumor. So a tumor is a collection of cells that have been transformed. Now, your tumors can be either benign or malignant. A benign tumor is a tumor that has not invaded any other tissues around it. It started with a skin cell, it stayed in the skin cell, it didn't invade any muscle or anything like that. And benign tumors can usually be removed with no problem. A malignant tumor is a tumor that has started to invade other tissues. So it started at one type of cell and then it began to invade other cells and start stealing blood supply. And it's not just in the skin cells. Now it's in tissue, or it's in bone, or it's in your lymph nodes. Once it starts to undergo, once it becomes malignant, it can also metastasize. And when it metastasizes, which you don't see here, that's when it actually begins to spread throughout the body. So you may have heard someone say it got in the blood or it got into their lymph system. And so you may have started off with breast cancer, but now it's spread to the lungs or spread to the liver or something like that. So that's when it has metastasized. Cancer cells do not respond to signals that normally regulate the cell cycle. They do not obey checkpoints. They don't need growth signals to actually start growing and dividing. They tend to either have an abnormal system that acts like it had a growth signal, or they make their own growth signals, or they make what's known as pseudo signals or fake growth signals. So cancer cells do not obey density dependency. That's how we get the tumor. They do not obey anchors dependency, and they also do not need growth factors in order for them to grow and divide. Every type of cancer cell is different. There's no one size fits all treatment. And so hopefully one of you guys one day will be able to research more, find out more, do more, and help eradicate cancer.